So if you just look at like nutrition labels, you might think protein's just like this generic globby thing, but actually proteins, there's a lot of different proteins, and they're like these molecular workers in your cells that can do all sorts of things. Like some proteins put together DNA, some proteins help hold together your cells, some proteins help move things around your cells. There's all sorts of different proteins doing different things, but your body needs different ones at different times. So you have all the instructions for making all of these proteins that you'll ever need in your DNA in the form of genes, but your body needs different proteins at different times, and it needs different amounts at different times, and so it has to control these levels of these various proteins. And I study this way that your cells help control how many proteins of different types are in your cells in this sequence specific manner. So it's this process called RNA interference, or RNAi, and it helps selectively prevent certain proteins from getting made, so you can help regulate the levels. And it's really cool because the sequence specific manner allows you to you can like artificially, so like as therapeutics or in the lab, if you want to see what a various protein does in cells, you can prevent that protein from being made and seeing what happens. So I want to tell you some more about RNA interference, or RNAi, and the protein at its heart, Argonaute, or AGO. And so here is just an intro that I've been working on, and that, so it's still a work in progress, and I know I need to change it before I can actually like make it work, but I wanted to give you a look. Each protein has its own genetic recipe that is present as a region of DNA called a gene. There's different genes for different proteins, but your body doesn't first go straight from the gene to the protein. Instead, it first makes a messenger RNA copy of those genes in a process called transcription. Then, in a process called translation, a protein-making complex called the ribosome moves along that mRNA, making the corresponding protein. The ribosomes can use these mRNAs multiple times. You could have multiple ribosomes translating an mRNA at the same time. Ultimately, the amount of protein that's made corresponds greatly to the amount of mRNA, to the number of mRNA copies there are. So cells can control the number of copies in order to help control the amounts of proteins that are made. So sometimes your cells might overshoot and make too much, or you might just not need that protein anymore in a new developmental stage, or that sort of thing. And so one of the ways that your body can control the levels of these mRNAs is through a process called RNA interference, or RNAi. So it can actually decrease the levels of specific messenger RNAs in order to decrease the amount of their proteins that are made. And it does this in a sequence-specific manner, so you can see it's only affecting the blue protein, the blue mRNA, and not the other two. So how does it do it? At its heart is this protein called Argonaute, or AGO, that I study. But AGO doesn't work alone. Instead, it works as part of a team with a small RNA, which could be a microRNA or an siRNA. We call it a guide. And when it binds to this guide to form this core RNA-induced silencing complex, or RISC. These small RNAs are about 22 nucleotides long, and at their 5' ends, they have a 6 to 8 nucleotide long seed sequence that allows AGO to use them as guides to find and bind messenger RNA targets that contain regions of complementarity, typically in their 3' UTR, so a regulatory region downstream of the actual protein-making instructions. After binding, alone or with the help of cofactors, as we'll see, they can induce translational repression, so stop the mRNA being used to make protein and actually get the protein degraded. And as a result, less protein is made. And the really, really key thing about this is that it's happening sequence specifically. So it's based on that small RNA that gets loaded. And so AGO can, in this way, be like programmed to go and decrease the levels of its specific proteins based on these small RNAs. And it can do this again and again to repress multiple targets. So where do these small RNAs come from? So the main small RNA that our bodies use in this pathway is called microRNA. And so these microRNAs have genes, just like proteins have different genes. But these genes are going to function as RNAs. They're a type of functional RNA or non-coding RNA. So instead of getting made into messenger RNA, they get made into microRNAs. But they have to go through a processing step, a couple of them actually. So when they get transcribed in the nucleus, they start as this primary microRNA. So it's this long hairpin, and then this microprocessor complex, so this complex of proteins, then chops off the top of the hairpin, so you get it's still a pretty long hairpin, too long for the purposes, but it's this longer hairpin, but it's chopped off from the base part, and this pre-mRNA gets exported out into the cytoplasm, so the general interior of your cells, and then it meets another pair of protein scissors called dicer. So dicer is kind of cool because it's like a scissors and a ruler in one, so it knows where it's going to cut to make it the right size for AGO. This is also where siRNA precursors come in. So siRNA stands for small interfering RNAs, and these typically come from exogenous or outside sources of double-stranded RNA. And so plants and invertebrates use this as an antiviral defense mechanism. So they use the virus's own RNA against it to target the virus. But mammals, we have evolved more complex immune systems that are multicellular and use this interferon pathway and all this other stuff. So we don't really use antiviral RNA, siRNA this way, but our cells can use siRNA that's given to us as like some sort of drug, or like especially in the lab we use it a lot for knocking down specific genes in a sequence-specific manner, because as we saw, it goes really great with that. So we still have the machinery because it's using the same machinery, most of it, but we don't use it for this like antiviral defense mechanism, at least not usually. But anyway, so those duplexes or that hairpin gets loaded into dicer, and then dicer is going to cut it into this 22 nucleotide long duplex with these overhanging ends, and that gets loaded into AGO to form the pre-RISC. So RISC is RNA and desilencing complex, remember? So right now the guide, so that orange strand, is hidden. And so remember, the guide is the part that has to match the target in order for AGO to bind and repress that target. And so we need to expose that guide by removing the passenger strand. And so AGO kicks that off, and now you have the mature core RISC. So now that guide is exposed, and it can go and seek out and to find and bind to messenger RNA targets that contain complementarity to at least that seed region, and often more extensive complementarity further down the RNA. Upon binding, AGO, if the target is fully complementary to the guide, so this is the case with like siRNAs, as well as some plant microRNAs, and if the Argonaute has this is a slicer, and so not all Argonauts have this activity, but our main Argonaute, AGO2, does. And what it can do is it can actually slice that target across in a specific spot across on the target, and then that target can get chewed away by exonucleases, leading to mRNA degradation, and you can't make proteins, so you have target repression. And if the target is partially complementary, so this is the case with most animal microRNAs, then AGO can't slice it, and it has to recruit cofactors. So it brings in a decapping complex and a deadenylation complex with the help of this scaffolding protein called GW182. So these cofactors help remove those protective ends so the mRNA can get degraded. It can also inhibit translation and sequester the mRNA so they can't get used. And then AGO can release the target and do this all again. So at the heart of all of this is Argonaute, or AGO, and so understanding how AGO works has helped us understand all of this, and that's what I've been focusing on these last few years.